But there's more to the biology and pathogenesis of leukemia than just the mutational profile in the leukemic cell itself. It's the microenvironment of the disease as well. Eunice, do you want to talk about that?
Right. I mean, I think that in the past, when we've studied leukemia, we've done it in a very preclinical way by taking the cells and putting them in cell culture dishes and adding drugs. Now, we've developed a lot of agents that way, but we think we sort of plateaued in our ability to kill leukemia cells in that way, because sometimes our results in the cell culture dish didn't translate into what we saw in patients. And it's important to remember that in a patient that the leukemia cells are living within specified niches in the bone marrow microenvironment, and those niches are very important. There's a significant amount of data that the adherence or adhesion of leukemia cells to endothelial cells within the bone marrow niche can mediate chemoresistance, just that particular microenvironment leukemic cell interaction. There's obviously a lot of emerging data that the microenvironment within the bone marrow space is inhabited not only by leukemia cells, but normal hematopoietic cells, but also immune cells, and that the leukemia cells are altering the local microenvironment to make it a more immunosuppressed environment, and that is important for the progression of the disease. And there's interactions even within in terms of oxygen delivery. Patients' samples are isolated in a laboratory, but patients' leukemia cells within the microenvironment are within an inherently hypoxic microenvironment, and the metabolic and mitochondrial changes that those cells undergo to allow them to persist and thrive within a hypoxic bone marrow microenvironment are very important. So I think as it's an emerging field, and as we'll be talking about shortly, there are development of novel therapies manipulating the microenvironment, manipulating adhesion, manipulating the immune microenvironment that now are starting to yield actual therapeutic implications.
Now, some of these mutations that we talked about will inactivate very important genes in normal bone marrow function. But you can also inactivate genes by epigenetic modulation, and that may be important as well in the pathogenesis. Jorge, do you want to discuss that?
Yeah, absolutely, and I think that's very important because it underscores how complex this biology is becoming as we understand these different elements as we've been discussing now. So it's not only whether there's a gene that's mutated in the microenvironment, but also how the expression of certain genes is regulated, and little by little we've understood that this is very tightly regulated in normal hematopoiesis, and very frequently dysregulated in these leukemia cells, and that leads to their behavior, their abnormal behavior, and development of these phenotypes, and which makes it also much more difficult to control because we need to attack it from different perspectives. So we have learned then that there's ways to regulate the expression of the genes, and that's what these epigenetic modulators do, and it's allowed us to understand how we can try to control this expression of lack of expression. If we silence a gene that is an oncosuppressor gene, well, then that's not a good thing, and they develop that. And we've also learned that some of the mutations that we've been detecting in some of these patients, actually what they do is not activate a certain pathway, but actually affect precisely this epigenetic regulation of other pathways that become very, very important. So sometimes you can regulate that directly, sometimes indirectly through these mutations, etc. I think we still need to understand a little bit better how you affect this, because of course we are able to now, we're starting to be able to manipulate this epigenetic expression of the genes, but it's kind of a global phenomenon. It's something where you silence, but we don't know what genes we can control, and we don't know how we can regulate some genes. So the
precision's not there.
Exactly. So we still have a ways to go, but at least we know now that is an important phenomenon that needs attention, and we're starting to work on that. Now we need to understand better and refine it more into regulate what we want to regulate, and not touch things that we may end up having a negative effect if we do that.
Yeah, a few years ago, Ken Figueroa actually divided the patients by their methylation pattern into different prognostic groups. And there was some overlap between the fusion genes and some of the genetics, but not perfect overlap. But you're right, how we intervened to overcome this and improve the outcome of patients, I'm still not certain how we can do that.